All right, so we're going to get started with our first lecture. All right, so this is the first lecture for animal physiology, and we actually jump right into muscles. The idea is hopefully you've taken a zoology class of some sort, so you have some basic idea of animals have muscles, as well as perhaps anatomy and physiology. If you've taken anatomy and physiology, this will be a good review. So we're going to focus on muscles. Muscles are a particular tissue that are actually unique to animals. When you think about the different kingdoms like plants and fungi and bacteria, of course, none of those organisms have muscles. Fungi don't have muscles, plants don't have muscles, but animals do. So that is unique to animals. That's actually one of the characteristics of animals, is the ability to move. And of course, move intentionally. Now, not all animals have muscles. If you talk about sponges, for instance, the most simple of animals, they don't have muscles. But most complex animals have some type of muscles. Most of the kingdom of animals has muscles, whether it be an earthworm or an insect, fish. These are all animals, and they all have muscles. Now, our first exam will be around the week of September 23rd. I will probably open it up pretty early, and you can pretty much take this test online. You will probably get a little bit more homework for this class than you would have had it been a in-class, face-to-face class. Because if you were doing face-to-face, we would have, face-to-face exams. And so you'll probably get a little bit more homework, and the exams will be more of an open book kind of affair. So these are the essay questions that are typically found on my exams that you need to begin working on. And I will treat them as homework assignment as well. And you'll see that under assignments in your Western Online. And so here are some of the questions that you'll have, and goals and objectives that I want you to get out of this class, this particular lecture series. And so I want you to be able to know the difference between skeletal muscle, smooth muscle, cardiac muscle, understand the sliding filament theory of contraction, understand and know the basic structure of a muscle, know and understand how skeletal muscle contracts and smooth muscle contracts. This is actually a question right here embedded I needed to fix. Let me do so. Well, we'll fix this in a little bit. And then, basically understand sacrum length. Anyway, we'll come back to this regularly. So what are the characteristics of muscles? Muscles are elongated fibers or cells. So muscles are a cell. They may have one nucleus, or they may have many nuclei. We call it nuclei for plural for a nucleus, so multiple nuclei. And it is usually filled up with fibers that are proteinous, obviously made of protein, that help in the contraction of the muscle. So muscles are filled with fibers. They may have one nucleus, or they may have more than one nucleus. Now, the thing about muscle physiology is those scientists tend to have their own nomenclature, and so they like to have their own prefixes for everything. So anytime you see the word myo, that should clue you in that they're talking about muscles, or myos, or sacro. So these are all terminologies that are kind of specific to muscle physiologists or physiologists in general. But these may refer to other parts of a cell, like a sacroplasmic reticulum would be part of the endoplasmic reticulum. but it's specific to muscle fibers. Or a saccharolima is actually the cell membrane of a muscle. So again, these are special terminology that tends to be associated with muscle physiology. Now, for complex animals like ourselves, we have three major types of muscles. We have skeletal muscle, cardiac muscle, and smooth muscle. So most animals will have a mixture of these different muscles, or at least most vertebrate animals will have that. And then you'll still see the basics of this in other animals. So an insect will have something that's like skeletal muscle, but insects don't have a skeleton. But the muscles are obviously meant for contracting and moving on their exoskeleton. They don't have an endoskeleton, but they have an exoskeleton, but it's obviously quite a bit different. The function of muscles are obviously primarily geared towards movement, trying to get around a room, trying to speak. Your hairs on your skin are moving in part due to muscles, or if you're trying to digest something that you've eaten, that is due to smooth muscles. So there's skeletal muscles that help you move, smooth muscles that help move your stomach and your intestines, your pupils in your eye. That's all examples of smooth muscle. And you can imagine cardiac muscle, of course, is the heart and arteries and things like that. So the function, again, is skeletal muscle to move around, cardiac muscle to pump blood, and smooth muscle to help in digestion and other aspects of your digestive system and circulatory system. Muscles are often obviously going to be important for posture and holding yourself upright, stabilizing joints. So it's not just the ligaments and tendons, but the muscles also help in stabilizing joints and movement. Muscles help in generating heat. When you're cold, you're shivering, and that helps to keep you warm. So muscles have all sorts of important functions. This here is a chart that tries to show you the differences and similarities between skeletal muscle, smooth muscle, and cardiac muscle. Skeletal muscle has what they call a striated appearance. In other words, you can see under a microscope lines, and it's the reason why you see all those lines is it's full of filaments that help in moving the arms or legs. It's going to be very thick, and that's why we call it striated. You can see this if you look at, maybe you have a piece of muscle that you're about to eat. You call it meat, or a piece of fish, which is still meat. Like some people like to pretend that it isn't. It is striated. It is muscle of an animal, and you can see the lines. Now you won't be able to see it quite at that microscopic level. And then we also have smooth muscle. Smooth muscle are thin filaments. They're usually very small cells. Striated muscles could be long muscle fibers. It could be the muscle fiber that goes up and down your bicep. Smooth muscles tend to be very small. And the interesting thing about cardiac muscle is it's somewhere in the middle. It's small, but it's striated. Now, the arrangement of muscles, and this will make a little bit more sense when you see some pictures, is we call them sacromeres. That is the what you'll see in a moment, which is what we call Z lines and filaments, or on how they're all attached. And we talk about the sliding filament theory. You'll learn about that more as we go through the different slides. Skeletal muscle is what we call voluntary muscle. In other words, I voluntarily moved my hand, my finger, I've thought about it. But again, remember that this is a little bit of an over exaggeration. I don't really sit and think about moving my muscles. So it could be unconscious. Or if I'm about something I touch something hot, I move my hand fast. That was relatively involuntary. So skeletal muscle, while we call it voluntary, there is an involuntary aspect to it. You'll find out later that calcium is very important for muscle contractions. Smooth muscle is involuntary. when you're digesting a meal, it's moving around, and cardiac is involuntary as well. The nervous system as actively has a synapse from the motor neurons, and we call that somatic motor neurons, and they are the ones that innervate a muscle and help it to contract. Now, there's still neurons in regards to the smooth muscle and cardiac muscle, but it comes from the autonomic nervous system, again, the automatic nervous system. Skeletal muscles, they say there's no hormonal influence, but we know that skeletal muscle would at least be influenced by some hormones, at least in growth, if you use testosterone, for instance. Multiple hormones are obviously involved in the smooth muscles. So if you're relaxed, it helps in digestion, and hormones involved in that part of the autonomic system will help in contracting the muscles and so forth. We'll get into the different hormones a little bit later. And then epinephrine is involved in the cardiac muscle. So again, skeletal muscles are going to be attached to bones. That's the word skeleton. Smooth muscles aren't attached to bones, and they make hollow organs and sphincters and so forth. That's what the smooth muscles make up. And then the cardiac muscle is involved in the heart muscle. The skeletal muscle is multinucleated, so it has more than one nucleus. Smooth muscle is uninucleate, so it has the elliptical look. And cardiac muscle is also elliptical, but it has striations. We'll get into this other stuff later, but there's T-tubules associated with skeletal muscle, no T-tubules associated with the smooth muscle, and the cardiac muscle has T-tubules. Again, that might not make a lot of sense to you, but it will make sense when we go through the different slides. Skeletal muscle is a fast contraction. Smooth muscle is a very slow contraction, often. And cardiac muscle obviously is somewhere in the middle, because you have your heart beating all the time. Skeletal muscle tends to be an all or nothing response. Smooth muscle is somewhere in the middle, and graded muscle is somewhere in the middle. Excuse me. And then, anyway, cardiac muscle is obviously autorhythmic. It's always kind of beating, and it can actually beat without your brain. You could actually pull someone's heart out, and there are groups of cells that would cause the muscle to still contract. So let's move on to the next slide. So here is a picture showing you the contraction speed of skeletal muscles, cardiac muscles, and smooth muscles. And you can see the skeletal muscle is a fast contraction. Cardiac is somewhere or intermediate, and the smooth muscle is a very slow contraction. Here is a picture of the different types of muscle. At the top, we have our cardiac muscle. On the left is the actual picture of the muscle itself. You'll see that it has one nucleus, and we call it intercladed because it is attached to each other. And we see that it's striated. That's what those lines are, where you got thick areas and thin areas. This is due to protein filaments that you can't see in this picture that give it what we call the striated look. The cardiac muscle, on the other hand, smooth muscle, on the other hand, is elliptical. See how it has this elliptical shape? You don't see the striation. See that it's all kind of clear. And over here you can see the smooth muscle. We also have skeletal muscle here that you can see striations. And you'll see that there's more than one nucleus associated with it. And then over here, we can see another picture from another textbook where you can see that there's striations. And then here's the real picture. If you look carefully, you can kind of see in the purple that there's striations. And then over here, we have cardiac muscle, where we have an intercladed disc. And that's like a zipper holding the cells together. And then on the right, we see the striations and the intercladed discs. That's what those pink thick areas are right here. And then our smooth muscle, again, is elliptical, and it's uh, single or uninucleated. Again, skeletal muscle is multinucleated. You can see the striations. It's voluntary, under conscious control. And it's bundled up in connective tissue that ultimately leads to tendons that attach to the bone. Again, it's voluntary, striated, and has lots of nuclei. Now, if anybody lifts weights, you may have heard the term muscle memory, where if you lift weights and you work out in the past, your muscles get bigger. And then you say you take a big rest period, a year or something like that, and you start lifting again. It is true that your muscles tend to grow faster and you get back to where you were quicker due to this concept called muscle memory. And one of the ideas is that when you were working out back in the past, you increased the number of nuclei in your muscles. And in doing so, you have the ability to, because what does the nucleus do? It causes the production of messenger RNA and ultimately transcription and translation with ribosomes to make more protein. So you put in, when you work out, one of the adaptations is to make and put in more of these nuclei. And then when you stop, your muscles may get smaller, but the nuclei are still there. And when you start working out, they can respond a little quicker to that stimulus. So there is something to the idea of muscle memory and being able to rebound from a workout. Anyway, that's kind of the gist of it. Irritable, that means you can give it electrical shock or stimulate it, and it will respond and cause it to contract. Muscles contract, and that's what causes your arm to move. So here's a picture showing you skeletal muscle that's attached to a tendon to a bone. And then you can see that there's these different connective tissue layers that surround the muscles. We can have an epimysium, that surround all the muscle fibers. And then each one of these muscle fibers is again going to be a muscle cell. So these cells are very long and thin. You can see some blood vessels associated with it. You see a paramysium that's surrounding these muscle fibers. And this is where inside of here you would have a saccharolima and a muscle cell. And each one of these is a sacromere. We'll get more into what a sacromere is in a moment. But you can see the line from here to here is what we call a sacromere. It is the contractile unit. But anyway, all these different bundles come together to attach to the tendon. And so here's a little bit more about how that is used. Obviously, breaking a tendon is a really bad idea because they don't heal very easily and it has to be reattached surgically. Anyway, here's some more things about the tendons and connective tissue that allows it to attach to the bones in the skeleton or cartilage. Some animals are actually completely cartilaginous, like some sharks, for instance, or all sharks, actually. Now, smooth muscles, again, are going to be small elliptical cells with one nuclei, and you'll notice that you don't really see any striations. And what they do is they form a hollow tube. And then this lumen is where the food can pass through. This is just one example of smooth muscles. You'll have smooth muscles used in a variety of different organs as well. But you'll see that they're going in different directions. If you were to slice a tube, like for your intestine, slice it, you'll see some of the muscles are going this way and some of the muscles are going that way. This allows for maximum contraction of the intestines. And then this helps the food to pass through the intestine. So anyway, that is the smooth muscles. Again, you don't see much striations. The contractions are slow. And that's what's mentioned here. It's the autonomic nervous system. Now, that doesn't mean that you can maybe learn to meditate or relax and influence the autonomic nervous system and can help control some of your smooth muscle contractions. But for the most part, it's completely involuntary. Smooth muscles tend to be in these sheets and often going in different directions. Often a hormone is involved in its contraction, and sometimes you can have more than one nerve involved or multiple nerves involved, while skeletal muscle tends to be just one nerve. The neurotransmitters might include things like acetylcholine or norepinephrine, and each one of these things influences whether or not the muscles contract or not. Acetylcholine, for example, causes smooth muscles to contract via what we call depolarization. And we'll explain more depolarization in a future class, but this is basically a change in the electrical charge across the cell membrane of the muscle, while norepinephrine causes hyperpolarization, which is the opposite and actually causes the muscles to relax and less likely to contract. And so that's what this picture is showing you here. This is an example of an experiment that you may do with your physiograph or in our labs, where we work with a rat, small intestines, and we apply hormones to it, like acetylcholine and norepinephrine. And if it's acetylcholine, the smooth muscle will contract, and we'll actually see the chart of the depolarization. That's what this is showing you here, changes in the cell membrane charge, electrical changes that become more positive. And then you see down here, the muscle is contracting. Remember, it's a slow contraction, and that's what is being illustrated in this picture here. And so if we have acetylcholine, the nerve impulses increase in contraction, or depolarization increases in frequency, and the smooth muscle contracts. And if we wash or we add norepinephrine, hyperpolarization occurs, so you don't see the depolarization, and the muscles relax. Now, we have a couple different terms for smooth muscles, whether they interact with multiple neurons or singular neurons. We have what you call a single unit smooth muscle when a single nerve, or it could be multiple nerves, but it essentially is only acting on the surface smooth muscles. And then you'll notice there's what we call gap junctions that join these other muscle cells together. So in a sense, all these muscles are talking to each other through these gap junctions. And so when, let's say, acetylcholine is coming down in green, attaches to a receptor, the cell depolarizes, meaning that the charge becomes more positive. And we'll talk more about that a little bit later in this lecture or the next one. And that depolarization then begins to travel through the rest of the cells. So what happens is this green neurotransmitter causes this smooth muscle to contract, and then that information travels through the gap junction, causing the rest of the smooth muscles to contract. So they do it as a single unit. That's the idea of the single unit. It's all these different muscles are attached and kind of function together. This is typical in what you would see in, for example, the small intestine. There's not a lot of individual control. There's not a lot of fine control. It's all kind of gross movement, large movements. Now we have what we call multi-unit smooth muscles, where each one of the muscles are directly innervated separately with the neuron. So here's, again, it might be more than one neuron or a single neuron. That's not the more important part. The point is that these muscles are separate from each other. And so that allows for them to contract individually related to the neurons. And what is the value of that? That allows for more fine control of the contraction. When would you need something like that? For example, in the eyes, when you dilate the pupils, and we'll get into more how the eye works in regards to the smooth muscles later. But that is what is happening in this picture, is that each one of these muscles are contracting by themselves. So a single unit, don't confuse the two. Single unit means treating multiple smooth muscles and they contract together through these gap junctions that allow them to talk. While single unit is actually individual muscles talking and doing things by themselves independently. Now we go to cardiac muscle, and you can see that the cardiac muscle, we see striations, and we see intercladed disc, and we see a single nuclei. And over here we can see the actual picture of it. Now, again, the striations can be seen easily, and we haven't talked a lot about what the striations and what those filaments are actually made up of. But they're actually made up of proteins we call actin and proteins we call myosin. And they're important in the contraction process, as you'll see in later parts of this lecture and next lecture. Again, the intercladed discs are like sewing the muscle cells together. So here's a muscle cell and here's a muscle cell, and they're sewn together. So when they contract, they're contracting individually. We also have a group of muscle cells that we call pacemaker cells. They're cells that are actually in charge of electrical depolarization. So literally, if your heart was pulled out of your body, it would still be able to beat as long as it was of a proper temperature and moist and stuff like that, due to these pacemaker cells. As the name implies, if you thought about a pacemaker, have ever heard of that, you know that you can surgically add a pacemaker to a body to cause the heart to beat. Well, the pacemaker cells do that naturally. And we'll talk about how that happens, but it's a depolarization event that occurs. So again, hearts do have the ability to beat separately from the rest of the body, though obviously when the heart is intact, the brain and parts of the brain stem are in charge of causing the muscles to contract. So here's our skeletal muscle, and this is where we're going to start talking more in depth of skeletal muscle and how that muscle contracts. This will be our model for explaining the rest of the muscle cells and how they contract. So let's take a look at skeletal muscle. You can see that it's made up of these different fibers, and they join to tendons, and all of them connect together. So we're going to take some scissors or a knife, cut some of the fiber out. Remember, it's surrounded by various connective tissues. And then we're going to pull out an individual muscle fiber. And then when we pull it out, you can see here's the individual muscle fiber made up of all these other muscle fibers. And inside it, so each one of these are a muscle cell. When I say single muscle, so a single muscle fiber is a single cell. So it can be really long and obviously very skinny or microscopically skinny. And it's made up of these fibers, and that's what you'll see over here. And they're repeating units that occur throughout the cell. And again, I'll show you other pictures and videos that help to put this together for you. But here's our sacromeres. And that's what we call these lines going from the blue line to this blue line. That's just what the artist drew it as. Obviously, it wouldn't be a blue in color, but these are the sacromeres. And then here's the individual nuclei that make up the muscle fiber. We also have the sarcoplasmic reticulum, and we'll get into more about what that does, along with the plasma membrane, which is the cell membrane. So again, it has more than one nucleus, so we call it multinucleated or multinuclei. So we're going to strip away this cell membrane and this focus in on these contractile units, since that is the bread and butter of how the muscle is contracting. You'll also notice the mitochondria that is associated with all cells, and particularly muscle cells. Their job is to make ATP, andesine triphosphate. This is the energy source needed for the muscle contractions. And so here is our sacromeres, and you go from Z line to Z line, and then we have our M band that's in the middle, and you have your actin in the, or excuse me, your myosin in the center. That's what this is here. And you have your actin on the outside. And then you got your Z lines. And this M band is actually proteins that help to hold these myosin together. So let's make sure you understand. These are actin, and I'll show you different pictures over and over. These are myosin, and these are Z lines. And together from Z line to Z line, we have what we call our contractile unit. That is a sacromere. And we also have a protein that's considered one of the largest proteins in the body, or the largest protein, called the titin filament. And it's kind of a springy unit that helps hold the myosin in place. So anyway, that is the, you can read these things here. I tend to focus on telling you, and then I want you to go back and read the slides to help you to appreciate what I've told you. So we've got bundles of myosin. The myosins are the contractile unit, and they function like little hands and are able to move with ATP. And that'll make more sense when I show you a video or more pictures. So here is another picture showing you a sacromere. Now, the one thing I want you to get from this, try not to get confused, is when you look at the bottom, you can see your sacromere. You got a Z line to Z line. You can see there's actin that's attached coming off of it. You can see there's myosin in the middle. And the thing that you're not getting from this picture is that, because it's 2D instead of 3D, is that actually all these arrangements are made in a 3D kind of way. So if you have your myosin here, and then you see the myosin is here too, but it's actually all over, and then you'll see there's actin surrounding it. So it's not this flat image. So it'd be like, you know, here's your, this is your myosin, my fingers representing my myosin. There'd be actin all the way around it. There'd be actin here, actin here, actin here, actin here, actin here. And there are a bunch of little fingers that can grab onto the actin. You can almost think of the actin as like the rope or the cables, while the myosin are like the gals or guys from CrossFit pulling on the rope in a tug of war contest, pulling the actin. And this will make a lot more sense once you see a video and some pictures. So let's talk about actin a little bit more. The actin filament is obviously, if you look at the upper left corner where my cursor is going, you'll see that it's kind of like these little, again, it's not blue in real life, but that's what the artists, the scientific illustrators made it look. So here we got the blue circles, and each one of these circles is an actin monomer or protein. So each one is an individual protein, and you can see it's kind of surrounding itself and wrapping around. On top of that, actin filament, or these actin monomers, is another protein we call tropomyosin. So you can see the tropomyosin. It's just kind of surrounding. And then on top of the tropomyosin is another group of proteins called troponin. So I guess at this point, I want you to realize, don't get too intimidated by the fact that there's a lot of big words that might be thrown at you for the first time. And the nice thing about video is you can go back and watch this video again. So you have your troponin, which is attached to the tropomyosin. And I'll explain what all this stuff does in a moment. And then you have your actin. Now let's look at the myosin. The myosin filaments are zoomed in over here to the right side, you can see it's a protein that's surrounding itself, like, like a cable. And then here's what we call the globular head. The myosin, remember, is like a finger. And the globular head would be the tip of the finger. And it's capable of moving with ATP like this. Or you can think of the myosin like a hand and the actin like a rope. Grab on and pull with the aid of ATP, androgen triphosphate. This will make a lot more sense again once we get to a illustration or a video of it, as well as a cartoon depiction of it, telling you the different steps. And again, remember, it's going all over the place inside the muscle fiber. You might have your myosin doing this, and then you have your actin all around it. And so the heads are grabbing all over the place, all the different hands. That is what causes the muscles to contract. Now remember, you got the Z line to Z line. If you look at the bottom, what happens is the Z lines come closer together. And then when the muscle relaxes, they go apart. That is the sliding filament theory. So here is another picture of the ultrastructure of muscle fibers. The cell membrane is called the saccharolima. The nuclei is called the nucleus, or the nuclei are obviously important for holding the DNA. What does the nucleus do? It holds the DNA, makes messenger RNA. That messenger RNA then is sent out into the cell, hooks up with a ribosome, and is translated into protein. So we have the nucleus. Its job is to hold DNA, make ribosomes, make messenger RNA. That messenger RNA leaves, hopefully you know this from some biology class, joins up with the ribosome, gets translated into protein, and that protein is what makes up this myosin, this actin, and just about everything you can imagine inside a muscle cell. The striations are due to myosin. So the dark striations are due to myosin. The light areas in a striation is just going to be some thin actin. And so you can actually see the striations move when a muscle contracts. So again, I should point out in a nucleus, when I say multi-nuclei, I'm not talking about 2 or 3 nuclei. I'm talking hundreds of nuclei that are associated with a muscle fiber. Here is another ultrastructural picture of a muscle fiber. And you can see the cell membrane is called the saccharolima. And inside the saccharolima are T-tubules that are made up of the cell membrane going inside the cell. So if you imagine having Play-Doh, and you push your fingers into the Play-Doh, the outside of the cell membrane, or outside of the Play-Doh, is now the inside. That's what the T-tubules are. It's essentially cell membrane going inside the muscle fiber. Surrounding the muscle fibers is the sacroplasmic reticulum. The sacroplasmic reticulum is a fancy term for the endoplasmic reticulum, but it's specialized in muscles to hold calcium. You're going to find out that calcium is critical for muscle contractions, and that'll make sense when we get to the next lecture. Looking at the illustration again, you'll see that the muscle fibers, here we have a Z, this is a sacrum here, Z line to Z line, and then we have the thick filaments, and we have the M band. It's not shown here, but we'd have a tightened protein holding the muscle fiber. Obviously, there's lots of mitochondria associated with a muscle fiber. That's important for making Anderson triphosphate. So these are the critical parts of a cell. Again, a saccharolima is the cell membrane. A special name we apply to muscles and fibers. So here's a picture of the T tubules going in. So the cell membrane comes in and becomes the internal part, and we call that a T tubule because it has kind of a T shape to it. And then you see that the sarcoplasmic reticulum is surrounding the muscle fibers, stores calcium in it, and then we have our, so basically this would be our sacromere, the contractile unit. You know, the saccharolima is on the outside, that's the cell membrane, not to confuse the two. And then you can see that the myosin heads attached to the actin, because the myosin heads are like little power, that's the engine, that's the hands and power that you're going to see in a later video. So that concludes our first lecture for animal physiology. Shortly, I will begin to talk about the sliding filament theory. Again, what are some of the key concepts that you should have gotten out of this lecture? Well, you need to know smooth muscle versus cardiac muscle versus skeletal muscle. You need to know what a sacromere is. You need to know what styration means. You need to know what a T tubule is, or a saccharolima. What does the mitochondria do? What does the nucleus do? How, you know, compare and contrast those different muscle fibers I talked about earlier. And that is the main objectives for the first lecture. I will shortly start the second lecture to begin to talk about how a muscle fiber contracts. Anyway, thank you so much for your attention. Please study hard and try to learn this topic. Ultimately, reading and learning can come from a lot of sources. I'm your guide to this, but don't be afraid to, actually, I guess I'm encouraging you to look at some YouTube videos, look at some online sources, whether it be your textbook or open book sources like your textbook. Look at Wikipedia. It'll actually provide you some decent information. Look at just about anything that looks like a credible source. Again, thank you for your attention, and I look forward to having you in this class.